Right, so one of the major research programs in biology today is trying to understand how all animals are related to each other. To do this, what we do is create an evolutionary tree where we put all animals in context with each other to figure out who is more closely related. This research has given us a lot of insight into evolutionary biology. For example, it's given us insight into the evolution of body shapes and sizes over time, and the evolution of interesting characteristics like the behavior patterns and interesting feathers of the birds of Papua New Guinea. This research has absolutely exploded in the last couple of decades, with our largest evolutionary trees being created yet. For example, in 2012, the Big Bird Tree came out, where they put every single known species of bird on an evolutionary tree. The Open Tree of Life project has an even grander goal of putting every single species on the planet on a tree. Part of the reason we've been able to build these huge trees is due to the fact that the accessibility of the data has increased. The data in this case is DNA. Where we used to spend weeks in the lab getting a single sequence of DNA, we now have the technology to get hundreds of millions of tiny reads of DNA from a single species. These reads generally come in the form of tiny strands of DNA, about 100 base pairs each making up a tiny fraction of the genome of the organism of interest. Now the big barrier to researchers is how do we sort through and computationally analyze the data to pull out the specific genes we are interested in. And this is one of my major research goals, is to build tools to help reduce that barrier of entry for researchers to deal with these large data sets. Often what we want to do is pull out a single gene or a bunch of genes from different organisms so we can look at those sequences and then build the evolutionary tree with those organisms. To do that, then, we want to sort through these large data sets and find those specific genes. Traditional methods require that we assemble all these little tiny reads into a huge genome before we can find the specific gene of interest. And this process usually takes about a week to do. So I created a piece of software that can help reduce that time. It's called aTRAM, Automated Target Restricted Assembly Method. And rather than assembling all the reads into a huge genome, what the software will do is go through that large data set and just target the specific reads that will assemble the gene of interest. In this way, we've reduced that time of assembly so that we don't have to assemble this large genome, but rather just target those reads and reduce that computational time. aTRAM is really easy to run. There are only 2 steps in this process. The first step is the sorting step. So what the program will do is go through this large data set and sort it into tiny little databases that the program can easily search through. The next step is the searching step. So what the researcher will do is tell the program, this is the gene I want, and the program will then go through all those little databases and find the reads and assemble the specific gene and hand that back to the researcher. This step can be done over and over and over to find many genes. So we wanted to determine how well the software works on a real data set. So we ran this on the adorable little critters that I study, which are sucking lice. And we had a database of 28 million reads, and we wanted to see how quickly we could assemble 1,500 genes from this data set. So we ran aTRAM using the sorting step first. So we sorted through 28 million reads, and the program took only about 1 hour and 45 minutes. So this process, which normally takes up about a week, we've now reduced to a matter of hours. We then wanted to assemble all of the genes from aTRAM. So we searched for 1,500 genes. Essentially, we ran the program 1,500 times to see how quickly it could assemble all of these genes. And on average, the program took about 1 minute and 15 seconds per gene. So this is really exciting, because as we have larger data sets and we want to get more and more genes or more and more data for each species, this program suggests that we'll be able to reduce that computational time to a matter of hours for pulling hundreds of genes. We wanted to determine that our results from aTRAM are actually similar to results using other methods. So we compared the sequences from aTRAM to those using other methods and found that they're about 98% similar. This suggests that our data set is comparable to other methods and can be used. So as we move into this large evolutionary tree era, we are excited about the possibility of using this program to help grow our data sets of species, data sets of DNA, and build larger and larger evolutionary trees over time. So, lucky for you, the program is available now. It's on this website here. It's an open source program, which means the code is freely available. Anybody can take that code and use it in other programs, or anybody can develop code and add additional packages to our software. So before I finish, I just want to thank the Illinois Natural History Survey for employing me over the last year. It's been a real honor and privilege to work here. And I also want to mention my fellow programmer on this project, Daisy Pang, who's a postdoctoral researcher at the University of British Columbia, who really helped pull together this software with me. Thank you.